Okay, hello everyone. Very happy to be here, and thank you for joining us for our Cell Press webinar on Spatial and Temporal Genomics in Cancer Research. I'm Miaozhi Cai, Senior Editor at Cell, and I'll be your moderator for today's webinar. Technology advancement allows us to dissect the spatial and temporal changes in genomics compositions, gene expressions, cell morphologies, and cellular environments during development and disease progression. All various data collections led to the emergence of spatial and temporal genomics as a new avenue of research. There is a significant interest in method development and clinical use of information for cancer diagnostic and treatment, as they may help to elucidate the basis for heterogeneity of cancer. So in this webinar, Miriam Merad from Mount Sinai, Fei Chen from Broad Institute of MIT and Harvard, and Stephen Wang from Yale University will discuss the directions of its further development. So before we get started with the talks, we have a few practical points. So each of our speakers will talk about 12 minutes. Following the talk, we will have a Q&A session that covers all three presentations. If you have a question for any of the speakers, just click on the Ask a Question button and then type it in. You can enter a question at any time. No need to wait until the end. We will try our best to pass on as many questions as we can. It is helpful if you can include the name of the speaker to whom you would like to direct your question. So before we begin, I would like to thank our speakers for making the time to join us today, and also our sponsor, OriGene, for supporting the webinar. So, and now, kicking it off is our first speaker, Miriam Merad. Miriam, thank you for joining us today.
Okay, so my great pleasure to be here always. Let me just make sure I'm sharing my screen. Can you guys see my screen?
Not yet.
Not yet, okay. Let me do this. Of course, even when you... Is it working now?
Yes.
All right, perfect.
Okay, sorry, I think we just lost Miriam, but we will have our second speaker to go first, Fei Chen, is with us now. So Fei, thank you.
Thank you for the introduction. I'm very excited to participate, and thank you for organizing. And really, as Mel said, the session I think is divided into both biology and also really the recent interest in new tools for our ability to map, basically bring genomics into the spatial context of tissues, and that's what I'll talk to you about today. Just a quick disclosure slide. And I think, I think there's no way to really talk about this without first bringing into the discussion of spatial single-cell transcriptomics. And I think one single-cell transcriptomics is one recent technological advance that has really revolutionized our ability to study molecular mechanisms in cells and tissues and in tumor biology. And that's because you can take a tissue and break it down into its constituent cell types and sample the transcriptome of each cell. And this is comprehensive, and it gets at the molecular programs that are driving the function of cells and the dysfunction of cells when in disease. But I think fundamentally we have to remember that to collect this type of data, we need to dissociate the tissue. And this is where we lose the spatial and temporal context about where cells come from. And I think that context is very paramount to studying basically how tissues are organized, how cells communicate with each other. And in particular, in cancer, it's very important for understanding the disorganization and heterogeneity of cellular structures, as well as maybe there's emergent multicellular niches and hubs, which are really important for, I guess, maybe the immune response or also tissue dysfunction, right? And so the other thing I think we have to remember is that cells and tissues are also very dynamic. They might migrate, they differentiate, they divide, and almost all of our tools that we have at our disposal today are kind of snapshots, right? We can freeze the tissue, or we dissociate it, or we lyse it, and we lose the context of those dynamics. I think those dynamics are very important to access. But today I'll really talk about my lab's work on spatial genomics, although we're very excited about the context of using molecular recorders like lineage tracing to study the context of dynamics within tissues. So today I'll mainly focus on a technology for assessing tissue scale spatial and spatial genomics, wherein you can retrieve the transcriptomes of cells across tissues. I think it's also very interesting to think about the structures of molecules within cells, such as the genome, and we're very interested in being able to do that as well. I think that'll be maybe the topic of the next talk. And so I think why are we interested in resolving tissue organization with spatial genomics? Well, I think we're motivated by three main classes of problems. The first is, as we are becoming so adept at collecting single cell transcriptomes, I think the most naive question is, like, where are these molecularly defined cell types located within tissues? And how does that organization relate to function? Beyond that, I think we're also interested in how are, within these cell types, what are the spatially differential gene expression patterns, right? And within a cell type, like, does its location in the tissue explain some of the variability in its gene expression? And what percentage or what aspects of its variability in gene expression can we explain? And then, of course, the lastly is, like, what are the gene expression changes with respect to, like, the pathology, right? We've known for a long time that we can go in and annotate tissue changes morphologically, such as in the case of H&E. But how do we connect those morphological changes with kind of the molecular mechanisms that we can discover through sequencing? So these are kind of the three motivating problems. Today, I'll really talk to you about how to address the loss of spatial context when performing single-cell sequencing by discussing a tool called Slide-seq, which enables genome-wide expression profiling in tissues at 10-micron resolution. And a lot of this work is, this is a collaborative effort between my group and Evan McCosco's, my neighbor at the Broad. And the big picture of Slide-seq is that it effectively lets you do untargeted single-cell sequencing. And at the same time, it lets you know where those cells came from in your tissue. And how do we do this? Well, in Slide-seq, we deposit individually barcoded 10-micron polystyrene beads in a monolayer on a surface. And the key is that in each one of these beads, there's millions of oligonucleotides with kind of a similar structure, a PCR handle or a constant sequence, a spatial bead barcode. What this is it's a clonal sequence for each bead that's different among beads. And you'll see how we use that in a second. A unique molecular identifier for counting transcripts, and a poly-DT handle for capturing mRNA. And what we do is we deposit these beads into a monolayer on a slide. And here's actually a cover slip. Here's a 40 millimeter cover slip with each one of these white circles is a 3 millimeter diameter array with about 100,000 of these 10 micron beads on it. And because these beads are deposited randomly, we then take a microscope and sequence the clonal bead barcodes for each bead in the array. I won't go into detail exactly how we do this, but basically we're trying to reconstruct the sequence of A's, C's, G's, and T's that make up each barcode for each bead. And we do that by taking a series of images where each color of one of 4 colors represents each base. So at the end of the day, we get kind of the XY locations of each bead barcode. So I think one of the main advantages of these spatial capture, spatial transcriptomic approaches is they're very easy to do once you have made the array. Here, once we made the array, it's very easy to do the experiment. You can take fresh frozen tissue and we section it directly onto the array, a 10 micron section. Then we basically perform molecular biology protocols that's very similar to single cell sequencing, such as reverse transcription and PCR. And we end up with kind of like a sequencing library. And this sequencing library is very much like what the libraries you get in single cell sequencing. For each bead barcode, you have a digital gene expression vector. So you end up a matrix of across bead barcodes. The counts where the columns are each gene and the counts of the gene that was seen for each bead. But now if you remember, we have the spatial location of each bead barcode, so you can just match them up. So you match up the kind of the sequenced XY locations of each bead, and now you have a spatial location for each read in your sequencing library. So what does the data look like? This is kind of actually one of the first experiments we ever did with Slide-seq. Slide-seq data has the same structure as single cell data. And what we can do is we can apply the similar computational tools such as dimensionality reduction and clustering. And actually here we ran Slide-seq on the mouse hippocampus. But in the very left, what we've done is we've just done unsupervised clustering and colored beads by the cell type they belong to. In the middle, we haven't performed any imaging. We've just colored the beads on the array by which cell type they belong to from the unsupervised clustering. And on the very right, you actually have an architecture of the tissue, and you see that we reconstruct the tissue architecture, but we also reconstruct the localization of cell types. So the takeaway is that there's no microscope. In fact, you've turned the sequencer into a microscope. And instead of just taking one image, you take many, many images across the transcriptome. And just some ideas about specifications. Slide-seq has very high spatial resolution, which we validated directly with single molecule FISH. And the resolution of the approach is about 10 microns, the size of the beads. It also, in some recent work, we've demonstrated that we can really improve the molecular sensitivity of the method, about an order of magnitude, to about 1,000 unique molecules per bead. This is similar to first generation droplet-based single cell approaches, and I think this can be improved further, just in terms of optimizing the molecular biology. But the nice thing is that because it's a spatial capture approach, it's actually quite scalable across many tissue types and organisms. And so we've applied it to many diverse mouse organs, a variety of human tumor specimens, as well as many developing embryo samples. And one of the things that we're focused on right now is how do we perform large-scale experiments in terms of batch effects and correlating these measurements across many sections in Z. And so the other thing that I think is very important with respect to all spatial transcriptomic data is that we want to know how to, there's two questions. One, we want to know how we can use single cell references that we've collected or we've already annotated cell types. And the second is that even though the beads are 10 microns, they're about the size of a single cell, there can be basically more than one cell captured per bead, about one and a half, like most beads have one, and some, like 30% of the beads have two cells. And what we actually like to know is we want to estimate the contributions from individual cell types to each bead on the array. And so we developed kind of a computational algorithm called robust cell type decomposition, which was recently published, led by Dylan and in collaboration with Rafael Irizarry lab. And this algorithm, basically given a single cell reference and a spatial transcriptomic data, can answer what combination of cell types and what proportion best fit each bead on the array. So now you can take a single cell reference and directly map it into space. This allows us to do a couple of things. One, it allows for really high resolution cell type mapping because single cell data sets are often much more diverse. And now we can project very fine subtypes directly into the spatial data. So for example, we've projected 27 interneuron subtypes into a spatial Slide-seq data set of the mouse hippocampus. And here you can actually see kind of very fine layered excitatory neuron subtypes projected into the cortex, and you can reconstruct kind of the layered architecture. And actually, RCTD we've demonstrated to work across many spatial transcriptomic platforms, including Visium. Although Visium obviously has much lower resolution than Slide-seq, you can reconstruct the same type of spatial architectures, and as well as imaging-based transcriptomics, such as MORPHISH. And so why is it really important to do this sort of cell type decomposition? Well, I think it's because there's been a lot of work focused on detecting spatially varying genes from spatial transcriptomic data. And in general, these approaches have not been cell type aware. But I think what you actually want to know in your tissue is that you want to know how gene expression changes as a function of space within a cell type, as a function of tissue organization. And so using kind of this sort of approach, we're beginning to be able to do that by discovering within cell type spatial variation, gradients within a cell type. For example, these two genes, which are within CA3 of the mouse hippocampus, or cellular neighborhood spatial variation. Like how does gene expression change in one cell type as a function of who its neighbors are? For example, here we discovered astrocytic genes, which change as a function of their proximity to excitatory neurons. Just for the last 1 minute of my talk, I want to mention that this sort of Slide-seq capture platform can really be adopted to many different types of sequencing modalities, including DNA. And in particular, in cancer, we might be interested in collecting multi-omic data where we integrate DNA sequencing data as well as RNA sequencing data to reconstruct kind of basically the clonal variation in combination with the transcriptomic variation. So we've adapted the Slide-seq arrays to capture DNA through kind of a transposase-based method in collaboration with Jason Bernarster's lab. And actually, it's easy to run serial sections because these beads share the same indexing. So now we can collect in serial sections DNA and RNA. And this just allows us to do de novo identification of tumor, chromosomal number aberrations, as well as clonality. For example, assigning subclones by lineage, by their relatedness in mutations. And then what we can do is we can kind of quantify the intrinsic and extrinsic factors of which contribute to gene expression. For example, if we have paired RNA-seq and DNA-seq data in spatial, we can examine basically like how does clonal assignment versus basically like environmental assignment affect gene expression. So here we can group basically genes by their percent variance in gene expression explained by either their clonal identity, like which subclone of the tumor it belongs to, versus like cellular neighborhood effects. One cellular neighborhood effect that we looked at was tumor cell density. And so we can assign like genes which are associated with subclone identity, and these might be associated with CNAs, and genes which are associated with, for example, tumor or immune density, which are more related to the cellular environment. So we think that's very exciting. I'm just out of time, so I just want to summarize that we've developed kind of very high throughput experimental approaches for high resolution spatial transcriptomic profiling. I think the development of computational algorithms is extremely important at this point for looking at cell type projection, differential expression, and cell-cell interaction. And we're really leveraging new tools in genomics to look at like multi-omics and something I didn't talk about, receptor sequencing in the context of T cells in space. So thank you, and I'm happy to take any questions at this point. Or actually, I guess we're taking questions at the end.
Yeah. Thank you, Fei. The technology is amazing. I look forward to more development and new biological insights from this. So we'll take questions at the end. Now, is Miriam coming back? Not yet. So maybe we move to our third speaker, Stephen Wang from Yale. So Stephen, thanks for joining us today.
Thank you, Mel, and thank you and Cell Press for the invitation. And it's really a great opportunity. I'm super glad to be able to report our work on studying spatial nucleome architectures from chromatin tracing and Mina. So as a conflict of interest disclosure, I'm a co-inventor on a patent applied for by Harvard University related to MERFISH. Our lab studies nucleome architecture. So I guess the first question is, what are nucleome architectures? We use this word to describe the spatial organization of the genome and other nuclear components. So we know the genome is spatially organized and folded across multiple length scales inside of the nucleus. First of all, DNA wraps around histones to form these nucleosome structures. And then on the other end of the spectrum, individual chromosomes actually occupy distinct nuclear space, and these are called chromosome territories. In recent years, thanks to advancements in sequencing technologies, we now know that at the length scale of several hundred kb, there are these topologically associating domains or TADs. Within TADs, there are often chromatin loops, such as promoter enhancer loops. And the TADs are further sorted into these A and B compartments. And A compartment is enriched with active chromatin, and B compartment is enriched with inactive chromatin. Other sequencing technologies also revealed that there are specific regions of the genome that are associated with nuclear laminar and with nucleolus, and these are termed LADs and NADs respectively. So these structures, they have been shown to control many essential genome functions, not just transcription regulation, but also DNA replication, mutation rate, repair, recombination, and also they show intriguing dynamics and changes during development, aging, and many diseases, including cancers. However, there are still many unknowns regarding the nucleome architectures in all these biological processes, largely due to technical limitations. Especially one key biological question is, what is the 3D folding path of chromatin at length scales above the nucleosomes in single cells? So from the TADs to the AB compartments to chromosome territories, our understanding of the nucleome architecture is largely blobology. So these are essentially larger and larger chromatin blobs. How do we get the real 3D folding paths of chromatin in single cells? And conventional sequencing technologies do not directly measure the 3D folding paths and often rely on population averaging, so they don't really answer this question. Conventional imaging techniques lack the resolution and the multiplexing ability to reveal the 3D folding paths. To tackle this question several years ago, we introduced this image-based 3D genomics technique called chromatin tracing that images and pinpoints the 3D positions of numerous genomic loci along the same chromosome and link them to reveal the 3D folding paths of chromatin. The challenge here is if you simply label so many genomic loci along the same chromosome, you don't know which one should be linked with which one. And also, due to the diffraction limit of light microscopy, their fluorescence actually are all connected into one patch, so you cannot even resolve the spatial folding, so many genomic loci. To tackle these difficulties, we designed this sequential imaging strategy. So we sequentially label, image, and pinpoint the genomic loci one after another until all of them are sequentially imaged and revealed, and we can link them based on their order on the genomic map. So this way, one can really reveal and resolve the 3D folding paths of chromatin along individual chromosomes in single cells. So this work really opened up this field called the image-based 3D genomics, and there are many following studies from many different labs, including several more works from my independent lab at Yale, applying this technique to different length scales and to different model organisms and biological processes. And these work are all based on actually multiplexed fluorescence in situ hybridization, or FISH. So basically a library of primary probes are simultaneously hybridized to the targeted genomic loci simultaneously. And these primary probes all have this overhang region that are unique to each genomic locus of interest. And then one can basically sequentially hybridize dilabel the secondary probes and image them and remove the signal before the next round of hybridization. So in such a way, image all the genomic loci one after another. But we also see the field is rapidly expanding to other varieties of technologies also, such as the very exciting in situ sequencing technique that Fei just introduced. Definitely check that work also. In our work, one thing that we focused on is to combine this highly multiplexed DNA imaging with highly multiplexed RNA and protein imaging all to in a single integrative platform called Mina, or multiplexed imaging of nucleome architectures. So in Mina, we achieved multi-scale and multifaceted nucleomic imaging across many length scales in mammalian tissue in a cell type specific manner in single cells. So in this work, for example, studying mouse fetal liver tissue section, we first look at the chromatin folding organization across 4 orders of magnitude of genomic lengths, all the way from promoter enhancer interactions to TADs, to compartments, to chromosome territory. And then we asked how are the chromatin folding differ among the diverse cell types in the fetal liver. And to answer that question, we imaged 137 different RNA species with a technique called RNA merfish, which is a multiplexed RNA imaging technique. Among these RNA species, 55 of them are cell type marker genes. And we also label the cell boundary, so we can segregate the individual cells and distinguish the cell types and answer the cell type specific questions of nucleome architectures. we also try to answer like how are the genome folding associated with gene expression changes. And to answer this question, the rest of the RNA species are actually from genes from the traced chromatin region. So we measure their expression as well. And remember, we're interested in other nuclear components as well. And in this work, we label the cell nucleus and cell nuclei so that we can profile the association of all these genomic regions with the nuclear laminar and the nucleoli and study those nucleome architectures. And finally, because everything is done in situ, we naturally preserve the cell positions in this complex tissue and also the signaling molecule positions, so we can study cell-cell interactions and the underlying signaling molecules in cell niche neighborhood. Here I'm showing several example images from the Mina platform, so from the same field of view in the same cells. I'm showing here the chromatin traces. If you zoom in on two of them, it looks like this. It's a mouse chromosome 19 folding trace. And then here are individual molecules of RNA, and each one of them is color-coded with over 100 pseudocolors to show their different RNA species, cell boundary labeling, cell type marker gene expression, cell type identification, and also the labeling of the whole nuclei and the nucleoli. Next, I hope to show several example analysis of what we can derive, what kind of biological questions we can answer with this high-dimensional multi-omic technique and data. The first, in fetal liver, based on prior knowledge, we already know that the hepatocyte cells express this very important metabolic gene called SCD2. The other cell types don't express it that much. However, if you look at the genome annotation, you'll see that near the SCD2 promoter, there are several enhancer clusters. Which one or ones of them interact with the SCD2 promoter in fetal liver hepatocyte is unknown. Using the Mina platform, we can basically group the hepatocyte chromatin traces and the non-hepatocyte chromatin traces of this region separately, and we can show that this enhancer interact with the SCD2 promoter. So Mina basically reveals nanoscale chromatin folding and can discover cell type specific promoter enhancer interactions in complex tissues. And then we asked, what about the larger scale chromatin folding? Particularly, how are the AB compartmentalization scheme differ in the different cell types in fetal liver? So we know that the TADs, the chromatin domains, they are sorted into this A and B compartments in a whole chromosome. And A compartment is enriched with active chromatin, B compartment is enriched with inactive chromatin. But these discoveries were made in homogeneous cell cultures. Now we have a complex tissue of so many different cell types. With Mina, we can profile the AB compartmentalization scheme and show that the scheme differ between the different cell types. We can also prove that in single copies of the chromosome, indeed, the A and B compartments do physically exist as separated chromatin regions. And the changes of the AB compartmentalization scheme, they are associated with gene expression changes. So Mina reveals cell type specific AB compartmentalization schemes that are associated with gene expression changes. Next, we branch into the other nuclear architectures. We ask, what is the relationship between AB compartmentalization and the nucleolar or laminar association of chromatin regions? Here, we're measuring the rate of the TADs to be associated with the nucleolus or nuclear laminar, and we compare that with the AB compartment score. We see predominantly negative correlation between these measures. However, you see there are systematic changes between the different cell types. So these nuclear architectures, they are cell type specific too. And we further show that this segregation of the AB compartments, they do not depend on the laminar or nucleolar association. So these results show that chromatin compartmentalization, nucleolar association, and laminar association do not solely determine each other, and they may offer orthogonal controls to genomic functions such as transcription regulation in a cell type specific manner. And finally, we are asking what are how the fetal liver cell types are arranged in space. And here we are projecting the cell type identities back onto the tissue image. From these images, we can analyze which pair of cell types are preferentially being neighbors with each other than expected. And we can see the particular cell types pairs are indeed preferentially being neighbors. Some of these discoveries are consistent with prior knowledge. For example, these red erythroid cells seem to surround macrophage, and this is called the erythroblastic island structure. However, some of the cell-cell interactions are unknown. For example, hepatocytes seem to be interacting with very short progenitors. And this is indeed shown in our tissue image as well. We ask what molecular interactions may underlie this arrangement. And from our hundreds of RNA species that we profiled on the same tissue image, we find a pair of ligand and receptor genes, KIT ligand and KIT, that are preferentially expressed in these neighboring hepatocytes and the erythroid progenitor cells. So basically, Mina can potentially reveal cell type interactions and the underlying molecular mechanism. As a summary, chromatin tracing allows direct spatial tracing of the 3D folding paths of chromatin in individual chromosomes in single cells. Mina enables multi-scale integrative nucleomic imaging in complex mammalian tissue and allows de novo discoveries of nucleomic architectures in a cell type specific manner, studying of the relationship between architectures and transcription regulation, also studying of cell-cell spatial interactions. This is because we now have one technique to image them all and in the darkness of the microscope room, bind them. And of course, our interest is not just limited in tissue development in hepatocyte, in fetal liver, for example. We have several works in the lab applying the Mina technique to different types of cancers, and I hope to report those results in the near future. And finally, I'd like to thank my lab at Yale and my collaborators, Professor Samuel Katz, Xinhua Pan, Sherman Wiseman. The work presented here were largely led by two talented graduate students, Miao Liu and Yanfang Lu. And thank you all for attending this presentation.
Thank you, Stephen, for bringing this data and technology from darkroom to us. Very interesting. I'm a big fan of Merfish, so I encourage people to put the questions in the chat box as well, so we will have discussion later on. So I think we have Miriam back with us. Very nice to have you back, Miriam. It's all yours now. I think you are muted.
Yes, I am muted. I'm very sorry, for what happened. So, can you tell me when did you guys lose me? Really at the beginning?
Yeah, very beginning. So you can start from beginning. So.
All right. Okay. Well,
yep.
I'm going to do that. I will try to be fast because I understand that we have some time to. We are on some time constraint here. Can you see my screen?
Yes.
I am anxious now to do something wrong.
No problem. Take your time.
Okay. Yeah. All right. Okay. So can you still hear me? You hear me, right? Let me try to put these things. Okay. I'm just trying to see whether I'm not going to do something wrong. Okay, so I was talking about myeloid compartment in tumor. I think you may have seen these, and specifically, I was emphasizing that macrophages are the largest immune cell compartment in tumor lesions, but there is very little known about this macrophage compartment because most of the biology of macrophage has been obtained in vitro, and there's very little knowledge of primary macrophage, especially in human lesions. I don't know whether I've been through this, but this is an important slide that you guys have to remember because this is starting to be part of the textbooks, but we now know that there is two main population of macrophage. We think they do things very differently, at least in some disease lesions. There is this big tissue-resident macrophage compartment that self-renew in tissue that is present prior than any type of tumor development. They are heavily imprinted by tissue Q. They play a key role in tissue repair. And then there is another population of macrophages that are recruited mostly in response to inflammatory cues. You guys, I'm still with you guys, right? Yes, you can still hear me. Okay. Yes. Just want to make sure, that...
I think yeah, perfect.
Tell me, you hear me well and the slides are okay? Okay.
Yeah, I'll let you know.
All right. And then there is this other compartment that is recruited in response to inflammatory cue. So tumor lesions, for example, produce a lot of inflammatory molecules, in fact, very, very early during tumor progression. And we have observed the reorganization of myeloid compartment already in the bone marrow that promote the release of myeloid progenitors and recruit myeloid progenitor in the lesions. And they also are really part of the tumor lesions. So the first things we did to really learn about the macrophage compartment is we performed this unbiased profiling using Slide-seq, which allows us to look at first the antibody profile of cells and their RNA program using this 10X platform. And we first decided that we were going to spend lots of time really profiling the immune microenvironment of treatment-naive tumors. We are focusing on few tumors, including lung cancer lesions. This is what I'm going to focus on today, but also HCC. And we are going to build this knowledge base and then build on top of it. Then we are doing same exercise in treated tumor, treated with monotherapy, combination therapy, etc. But today I'm going to talk about our effort to profile these treatment-naive lesions. And here, so we use then prior knowledge to look for cells that express the same antibody and the same RNA program. And we use prior knowledge to call them NK cells, ILCs, T cells, or macrophage. And we identify several macrophage programs. Then we look where they are distributed, because here, in collaboration with our pathologists, we always isolate a tumor and a tumor-free adjacent tissue. And then we purify the CD45 compartment from both the tumor and adjacent. Now we are doing it at different parts of the tumor to look at heterogeneity of the lesions. And then we do single-cell suspension and the Slide-seq analysis. So the first analysis we do is look at distribution of all these molecular and biasly identify molecular and antibody profile of these myeloid cells in the tumor versus adjacent tissue. And we spend a lot of time doing this. But then, because we want to know a little bit the contribution, of course, and the spatial distribution and the function of this compartment, we have to have access to some experimental model. So we have a model that of adenocarcinoma lesion that we like a lot in the lab that was developed by Tyler Jacks, which is this KP line, which expresses the KRAS activated mutation and P53 deletion. And these lines, when injected IV or intratracheally, form these nice adenocarcinoma lesions in the mouse and in the lung of the mouse. And the first things we did is use exactly the same profiling, so Slide-seq method, to look at myeloid program of these lung cancer lesions in mice. And then we use this cross-species analysis to look for programs that are identical in mice and humans. And we identify 4 programs that are similar. Here we coalesce different clusters that I've showed you before. And we are going to focus on these 4 different programs because we were able to somehow find, especially using module analysis. I don't have the time to go deeply in the analysis we performed, but we think that these are programs that are somehow engaged by tumor cues quite similarly in mice and humans. So now when we have this program, the first question we ask is, what is the origin of these myeloid compartment? Are they tissue-resident macrophage? Are they derived from blood circulating progenitors? And to do that, we use a fate mapping model that was developed by Boris Reises, and that we helped, in fact, profile. In these models, what we use is we use this molecule called MAP17, which is expressed on early adult hematopoietic precursors. We developed this mouse model that allows us to genetically label both the MAP17 cells conditionally. Upon tamoxifen induction, we conditionally label MAP17 and all the MAP17 progeny, and the genetic traces remain there completely untouched for the life of the animal. So we fate map the mice, and then when the mice are 6 months old, it takes a lot of time to really fate map faithfully the hematopoietic derived cells. We inject the tumor, and then in the tumor, we purify the labeled cells from non-labeled cells, and we look at the molecular distribution of this myeloid program in labeled versus non-labeled. And what we found is there was one specific cluster that was never enriched in labeled cells, which we call cluster 1. And this enabled us to really identify these cluster of macrophage as the tissue-resident macrophage. So these are the tissue-resident macrophage that are present in the tumor lesions. So the first thing we did is identify marker that allows us to identify them using flow cytometry so that we can probe their function, or also look at their spatial distribution. And what we found is that this cluster express very high level of CD169, high level of CD206, and this enables us to look at the spatial distribution of tissue-resident macrophage versus these adult bone marrow-derived macrophages. And what we found is that the tissue-resident macrophage, surprisingly, are always present outside the tumor lesions. And this was also the same in human lesions. Again, here we use this molecular analysis, we use the cross-species analysis to say, okay, these macrophages resemble the tissue-resident macrophage that we analyze using this fate mapping model in mice. So we think they also were enriched in both CD206, but also were enriched in CD10. We use these two molecules to profile now the tissue-resident macrophage, and we found they are abundant outside the tumor lesions. That was surprising to us because the tissue-resident macrophage are part of the lung tissue. I just want to make sure that you guys are here. Okay, you are here, and then so we then started to say, well, okay, so let's really look at the temporal distribution of macrophage during tumor growth, and we started now to image, in fact, the tumor lesion at different times during tumor progression. And what we found is that these tissue-resident macrophage are the first, in fact, to interact with tumor cells. This is what I'm showing you here. In red, these are the tissue-resident macrophage, and in green, these are KP cells, the tumor cells expressing green fluorescence proteins so that we can track them. So these are the first interaction is with tissue-resident macrophage. So the experiment we did is to probe whether this macrophage play a role at all in tumor progression, and here we use the reductions model in which we co-culture tumor cells with tissue-resident macrophage or monocyte-derived macrophage, which we found are abundant in advanced tumor lesions. And we ask whether they play a different role in promoting tumor invasiveness. And strikingly, what we found is that the tissue-resident macrophage were very potent at inducing this epithelial mesenchymal transition, where they reduced the expression of an epithelial program, induced the expression of a mesenchymal program, and induced the formation of this bridging colony. So they promoted really the capacity of macrophage to invade, in fact, the matrigel. I'm going quickly here because this paper has been published, but I just want to oppose, in fact, the ability of this tissue-resident macrophage to induce, in fact, EMT, this EMT program. But what we also found is that these tissue-resident macrophage were also interacting, especially early during tumor progression, with T-regulatory cells, and they were T-regulatory cells that I'm showing here in red. And this early, in fact, interaction was very intriguing to us and suggesting that maybe they were also inducing a T-regulatory cell program. And indeed, what we see is that there is a first wave of CD8 T cells in most tumors that we've looked at, and then the CD8 T cells are dampened. And I think this is, and we think that this induction of Treg by TRM is contributing to inhibiting this first wave of T cell accumulation. So the next experiment we did is we depleted this tissue-resident macrophage without affecting the monocyte-derived macrophage compartment, and we asked whether this will reduce, in fact, tumor invasiveness and T-regulatory cell induction. And this is exactly what we saw, is that in the absence of tissue-resident macrophage, tumors have difficulty to invade, despite the fact that the seeding of tumor cells was not affected. And we also saw that there was a reduction of Treg, but also a reduction, an alteration of the functionality of T-regulatory cells and an accumulation of CD8 T cells. But this TRM really, if we now depleted TRM later during tumor progression, there was no effect at all on tumor growth. And this is because the CRM are present, are as I've showed you, excluded then from the tumor foci. So in order for the tumor to continue to grow, well, no, not in order. I mean, something happened, and we are looking into exactly what's happening. We believe that the TRM are then excluded because they are going to form somehow or build a little stromal structure that's going to be harnessed by the tumor to recruit, and they are going to recruit monocyte-derived macrophage, which then become abundant in advanced tumor lesions. So we started then to look at this monocyte-derived macrophage, and indeed we saw that in advanced lesions, monocyte-derived macrophage dominate the tumor microenvironment, and the TRM are outside the tumor microenvironment. However, this monocyte-derived macrophage that we identify with our fate mapping models are also quite heterogeneous, but there was one program that was specifically abundant in the tumor, and this program expressed very high level of this molecule called TREM2. Now, these TREM2 positive macrophage, we have, heard about them mostly. So this is what I'm showing you here. In these human lung cancer lesions, there is abundance of TREM2 positive macrophages, and this is a mouse lesion. There is also abundance of TREM2 macrophage. Okay, so we had heard about this TREM2 positive macrophage. Before I go to here, I'm just showing you something. Is that this TREM2 positive macrophage, this molecular cluster that we identify in human, was also present in mice. And in fact, the program was quite identical. There was many transcripts that were identical between mice and human. We also looked at the temporal accumulation of this TREM2 positive macrophage, and what we saw that in contrast to the TRM that interact with tumor cells quite early during tumor progression, the TREM2 positive macrophage are recruited later during tumor growth. Right? So now we have these two programs that are shared between mice and human. They are abundant in advanced lesions. Now we are going to look at how these TREM2, we have now a tool to look at the role of this TREM2 positive macrophage in tumor growth. But this is what I've been trying to tell you, is that we have heard about this TREM2 macrophage mostly in the context of Alzheimer's disease, where TREM2 variants have been shown to be associated with increased susceptibility to Alzheimer's disease. We have also identified abundant TREM2 positive macrophage in colon cancer lesions. In fact, in a paper that we published in Cell, TREM2 positive macrophage had been found in cirrhotic liver. They had found also in adipose tissue, especially in obese patients. And they have been also shown to be abundant in experimental sarcoma cancer lesions by Marco Colonna and Ido Hamid recently, in fact, in back-to-back papers published in Cell. So this clearly, it seems that this TREM2 positive macrophage seems to accumulate in response to different inflammatory cues. Something that is common with all these programs is the abundance of either fat tissue or at least antigen cargo. And indeed, TREM2 is a scavenging receptor. So the next question we ask is whether this TREM2 program was induced upon uptake of tumor cell debris. And to ask this question, we purify macrophage and bias. Now we just take a myeloid marker, in this case, CD11B, and that are express high level of GFP or low level of GFP, and then we look at their molecular program. And what we found is that this TREM2 positive, the TREM2 molecular program that I described to you, was very strongly enriched in these GFP high macrophages that we isolated from mouse tumor lesions. Then we did now another experiment to further prove that this uptake of the cargo was inducing this program. We derived bone marrow-derived macrophages in vitro, and then we fed them with KP cells expressing GFP. And what we saw is that this feeding process really induced this program that includes the expression of TREM2, SPP1, GPNMB, ApoE, IL-7 receptor, PLIM2, all these programs that seems always to be co-expressed together, both in mice and human tumor-associated macrophage. And now we see is enriched in macrophages that have cleared or captured apoptotic tumor cells. Okay, so the next experiment we ask is whether what happens if we now try to delete TREM2, will we affect tumor growth? Marco Colonna had shown that deletion of TREM2 reduced the progression of sarcoma cancer lesions. So here we did the same experiment where we deleted TREM2 from the host, from the microenvironment, and injected this adenocarcinoma line. And we saw that in the absence of TREM2 positive macrophage, there was a very strong reduction of tumor growth. And this was associated with a reorganization of the myeloid program. We saw a reduction of this MOMAC cluster, an expansion of monocyte, an expansion of tissue-resident macrophage, here in blue, but also an expansion of a specific subset of DC1. And this seems to be very specific to this compartment because DC2, which are usually more abundant in tumor, was not affected by the TREM2 deficiency. So something was happening in the myeloid compartment of TREM2 deficient animal. Now, the problem is when we saw that there was such a strong tumor reduction, that's super cool, but the reduction of tumor burden could also contribute to all these myeloid changes that we observed. So we did a cool experiment, I think, where now we reconstituted mice with bone marrow cells that are with mixed bone marrow cells with a mixture of TREM2 proficient and TREM2 deficient bone marrow progenitor, which will give rise now to TREM2 proficient and deficient macrophage in the same compartment. Now there is no effect on tumor growth, so we are going to purify TREM2 positive and TREM2 negative macrophage because we label them differently genetically, and we are going to compare their programs. So here, the tumor cues are identical. We are just looking at cell and transient role of TREM2. And what we saw was very exciting. So first, we saw that TREM2 positive and negative macrophage uptake the same amount of tumor antigen. So the TREM2 was not really regulating uptake, but we saw that TREM2 regulated very strongly the inflammatory program. It's very strongly dampened inflammatory production and expression of co-stimulatory molecule, production of lot of inflammatory molecule, including the production of IL-15 and IL-18, which are very important molecules to activate and expand NK cells. And I'm going to show you. They also the TREM2 wild type was important for the expression of a lot of scavenger receptor and also a lot of checkpoint molecules, including this LILR beta 4, which we are very excited about. Okay, so that's quite interesting. So now we go back and ask what happened in the environment of this TREM2 deficient animal or TREM2 deficient animal. And what we saw is there was a very strong expansion. So I showed you the reorganization of the myeloid compartment. What we also saw is there was a very strong expansion of NK cells. And we are very excited about this because NK cells are always depleted from the tumor lesion. And we have seen that in human lung cancer also, there was a very strong depletion of NK cells. But also we saw an accumulation of activated NK cells. We also saw an accumulation of CD8 T cell, but also an accumulation of CD8 T cell that express XCL1 or express also granzyme B, suggesting that these were effector CD8 T cells. So the next experiment we did is we depleted CD8 or NK cells and asked whether we abrogated this TREM2 beneficial anti-tumor effect. And this was indeed the case when we depleted NK, but not so much CD8 T cells. So we are quite interested in these results. Also, it seems that the TREM2 beneficial effect was mostly due to the recruitment of NK cell and their activation, and we also abrogated this DC1 accumulation here. So this is quite interesting because it's the first time really that we are able to expand NK cell in a compartment and expand DC1. And because of this result, we are starting now a first in human TREM2 blockade study in advanced cancer lesions in patients at Sinai. And this will be done with a Pioneer, a small biotech that have been recently acquired by Gilead, a biotech that was in fact funded by Max Krom. So we are very excited about this. And in conclusion, I'm just going to summarize what I've showed you. I've showed you that tissue-resident macrophage promote tumor invasiveness through the induction of an epithelial mesenchymal transition program and induction of early Tregs that dampen tumor immunity. This is very relevant for cancer in situ lesions. We have shown similar results in breast cancer many years ago. There was a recent study showing that tissue-resident macrophage are also promoting this early tumor invasiveness in glioblastoma. So this seems to be really the property of this tissue-resident macrophage. This is very interesting to us because we have a very strong lung screening program at Sinai, which can detect these early in situ lung cancer lesions. And we are going to now start a clinical trial to try to activate or deplete tissue-resident macrophage without operating on this lesion and see whether we can somehow reduce tumor growth. I've showed you that the TRM are depleted from advanced lesions, which are dominated by monocyte-derived macrophage. So that provides us with a way of targeting monocyte-derived macrophage while sparing tissue-resident macrophage, which also have a lot of homeostatic property, including in the lung. They are the ones that are clearing the surfactant. They contribute to lung tissue integrity. So if we don't have to target them, then let's don't do that. I've showed you that TREM2 is a very good target of this monocyte-derived macrophage. I've also showed you that sensing of tumor cell debris induce a TREM2-induced program, and these programs contribute to the depletion of NK cells and DC1 from the microenvironment. And and that TREM2 blockade. So I've showed you the TREM2 deficiency, but in collaboration with Marco Colonna, we are also using a TREM2 antibody blockade, which led to very similar results where we can reprogram the myeloid landscape. I didn't show you the molecular program of TREM2. No, I showed you that the single cell sequencing of TREM2 deficient macrophage. You've seen them. So they now start to be inflammatory. They start to produce inflammatory molecule, recruit NK cell in the tumor lesions, suggesting that we can really reprogram their ability to promote anti-tumor immunity. And we think that this is really a very strong avenue to enhance anti-tumor immune response in lung cancer patients. These are my people. I know we are out of time. I'm not going to go through all the names here, unfortunately. This is our group wearing our vaccine campaign T-shirts, and I'll be happy to address any question at the end of the talk. I hope I didn't go. I hope it didn't take me too long. I'm done with my presentation. I hope that you heard me well.
Yes, beautiful talk, Miriam. It's very exciting discovery.
I'm glad that I was able to finish. Did I go over the 12 minutes too much or not? I didn't have a timer.
No, it's fine. But I think we are running out of time. We have a lot of questions in the list. So I know that Fei needs to go. Fei, do you have time to take one question?
Yeah, of course. I can take a couple of questions right now.
So first question for you is how do you quantify the density of tumor cells and stroma components in the tumor microenvironment from Slide-seq data?
Right, and there what we did was we used the transcriptomic signatures to map the cell types into the space, and it's a spatial density based on the transcriptomic signatures. Thank you.
I should stop sharing my slides, sorry. Okay, I should stop sharing.
So for our speakers, if you have questions to each other, feel free to interrupt me to ask.
Did I end share here? No. Okay, perfect. Well, I was going to ask my colleagues that I would love to now look at macrophage T-cell interaction in the microenvironment, right? And you know, we started to look at PIC-seq with IDO. I didn't show this. So we will purify myeloid doublet cell that express myeloid and T-cell compartment. And what's clear in the microenvironment is you have an enrichment of macrophage T-cell interaction that dominates, you know, over DC T-cell interaction. However, this is not the case in TLS. And so can we? So you know, to us somehow of targeted sequencing is the best way to go. At least initially. Can we do some target-seq where we just at least locate, you know, this myeloid T-cell interaction locally and then go deep?
Yeah, I was going to ask that as well.
Possible in today's world. You know, there's many ways. It depends on if you want to ask questions about. So, like, it depends on what your questions are, right? Like, are you interested in the differential expression of genes as a function of the interactions? And you probably already have a list of targets from single cell.
Yes.
In which case, it sounds like it's a good experiment for targeted imaging-based methods, as well, like, you know, like MERFISH or in situ sequencing, which is some of the technologies that Stephen talked about and I didn't get a chance to talk about, but that's great for targeted methods. If you're interested in doing discovery of what the transcriptome, like what transcriptomes are changing as a function of interaction, then you can use like untargeted methods like Slide-seq or spatial transcriptomics.
The problem is the resolution, right? So for us, it is important to start having a sense of single cell resolution. Just because I've showed you, right? Many of these genes are present everywhere, right? So, so tell me a little bit about the resolution that you guys have now. If let's say we want to go.
Stephen can talk about MERFISH more than anyone probably, but I think the resolution is very high if you know what the targets are.
Yeah. I started the collaboration with Murphy, Stephen, as you know. So I'm looking forward to very exciting results. I gave them 300 molecules that I'm very excited about, and they're all encompass all these macrophage compartments that I described here today. So I'm looking forward to these results.
Regarding the resolution, actually, I have a question for Fei as well. So indeed, the Murfish have this single molecule resolution, but I think for a lot of people, actually, they don't need to know the subcellular distribution of RNA, and maybe many people just wonder, like, a whole cell level, spatial resolution is enough. And in your Slide-seq technique, definitely, if you use the smaller and smaller beads, the spatial resolution would be better and better. But I guess the capture rate of the RNA will get lower and lower. So what's your estimate of like how small the bead is optimal, like a balance between the two? Or would you say your current size is already the optimal?
That's a good question. I think probably optimal is, as you said, basically the capture rate scales with area, right? Like there's just less RNA. RNA scales with area. And so if you have the size of the beads, you get a 4 times reduction. But then you can dynamically re-aggregate the beads if you have good computational algorithms. That's like a little bit like a different version of the segmentation problem. I actually think that probably the optimum lies at like 5 microns maybe.
Which you already achieved, right? Essentially,
it's close to where we are. I mean, we chose 10 for good balance between those things.
Yeah, but that is going to be impacted by how crowded, right, a place is, right? So there's heterogeneity in these tumor microenvironments. So we find that, for example, where there is lymphoid aggregate, resolution that you will require would be, much higher than regions that are more sparse, of cells. So it's important to continue to think about that the distribution is not equal, and that resolution is going to be affected by that.
Yeah.
Okay. I think we probably need to end this session, but it's great to see you guys are already talking about this, and then more and more questions are coming up. So it's great to have biology and technology all coming together, and then we can bring cancer research or many other biological research to the next level. And we also have many questions in the our question box. I'll send those questions to our speakers so that they can go through them and probably get back to you. With that, I think we have come to the end of our session. We would like to say thanks again to all of our speakers, Miriam, Fei, and Stephen, for their engaging presentation. We are very grateful to our sponsor, OriGene, for the contributions that made this webinar possible. We would also like to thank you, the audience, for tuning in today. If you missed anything during this webinar, or you would like to listen it again, the recording will be available shortly on thecell.com. If you have any comments about the webinar or suggestions for future topics, we would love to hear from you by email at cpwebinars@cell.com. Thank you. Bye-bye.
Thank you, Miao. Thank you for having us.
Thank you.
Yeah.
Bye-bye.
Yeah. Bye-bye.
Bye.